driving forces for molecular motion. So we're trying to develop an intuition for what governs the motion and the forces that propel molecules through a solution. And that provided us with a segue into our discussion of our first tissue type, and that's the epithelial tissue that provides a barrier between our internal physiology and our environment. And we talked about these very sort of simple models for electrochemical gradients, illustrating how you have both the electromotive force and chemical diffusion sometimes at odds with one another across a membrane. And we also talked about how if you have transmembrane proteins in a cell membrane, that provides the opportunity to control some of this flux. And if you actually want to repel an ion against a gradient, like its chemical concentration gradient, then that's going to require energy. And that's done by this transmembrane protein, which we'll talk a lot about. It's a carrier protein. It's called sodium-potassium ATPase, and it requires energy in the form of ATP in order to move both sodium and potassium against their chemical concentration gradients. We have another class of transmembrane proteins called channel proteins, which take advantage of chemical diffusion in order to move potassium, for example, across a cell membrane. And this is facilitated diffusion because that driving force of chemical diffusion is driving potassium across the membrane, then no ATP is required. Okay, so let's consider epithelial cells. We talked about this basic structure last time. You have a whole bunch of cells that are lined up. There are chemical barriers that prevent chemical diffusion between the cells. There are structural connections that create a sheet of cells to provide some structural integrity in this barrier. And we're going to consider our first example of epithelial transport. This is the control of something across the cell. Okay, guys, I'm going to stop because we don't want to have cell phones and we don't want to have laptops in class. Okay? So this is a non-technology. This is 1 hour when you guys are free from technology. You get to use just pencil and paper, old school. It's a great way to absorb the material in lecture. Okay, so if the goal of this cell is to control the flux of, in this case, a molecule from the environment. And when you, again, swallow something and it's in your gut lumen, it's really still in the environment. It's not until it crosses an epithelial barrier that it really enters your body. And so we would like to control that process so we don't just absorb everything we swallow. And also, so that we can maybe move, if we just depended on chemical diffusion, then what would happen is your bloodstream would reach the same concentration as in your gut lumen, which means you wouldn't be able to extract very efficiently the nutrients from your body. So instead, we would like to grab as many of the nutrients that we swallow, that we want to enter into the body. And so if we have control over the molecules that are absorbed, then it provides the opportunity to have a higher concentration of nutrients in our body than in the gut lumen. Okay, so those are the sort of large goals here in this very detailed process of getting a good thing, in this case it's glucose, from the gut lumen, which is what's on the right. So the apical side, or the top side of the cell, is this sort of Bart-Simpson shape here. And then on the basal side, that's where we're going to have this interstitial fluid and the nutrients are free to diffuse into the bloodstream. All right, so let's take the cell and rotate it 90 degrees. And that's what we have here. So up top is the gut lumen. Down below is our interstitial fluid. And we're going to focus on a single epithelial cell here, and we're going to consider all the molecular players that are involved in getting a glucose from the top to the bottom. Got some feedback. Okay, so for reference, apical surface top, basal surface bottom. And we abbreviate glucose as GLU. We want the glucose to go like that. We want it to enter into the bloodstream. And there are a couple of problems here. So first of all, there is a glucose chemical concentration difference between what's in the gut lumen. And in this situation, it might be far along in your GI tract where a lot of glucose has already been absorbed. But under this condition, we have a higher concentration of glucose in the interior of the cell. The cytoplasm has a higher concentration than you see in the gut lumen. Okay, so that's a chemical concentration difference that's problematic if you were to rely purely on chemical diffusion, right? It wouldn't go that way. Instead, the glucose will tend to want to remain in the gut lumen, where the concentration is lower. So that flux that we're trying to drive against a concentration gradient is going to require energy. And so where does the energy come from? That's the sort of key problem in devising a way to allow for this transport. Okay, so sodium potassium ATPase is going to make many, many appearances. It's prevalent in all cells, and it plays a role in this transport process. And again, it's just constantly churning along, hydrolyzing ATP, and pumping both sodium and potassium. And in that process, it's establishing a chemical concentration gradient for both of those ions. All right, so it looks like that. Now, so now we can add the concentration, in this case of sodium. It's going to be relatively high on the interstitial fluid side of the cell, on the basal side, compared to the interior of the cell. And the same is true on the apical surface as well. We've got a lot of sodiums floating around in the gut lumen at a higher concentration than in the interior of the cell. Now that sets up a solution to our problem. Okay? The problem is getting glucose to move against its chemical concentration gradient. And that is achieved by taking advantage of sodium as a source of energy. So sodium potassium ATPase creates these concentration differences. They want to go in the opposite direction. Sodium wants to rush inside of the cell. That's just via the magic of chemical diffusion. Okay, we've created the concentration difference, and now the driving force in the opposite direction is chemical diffusion. And so you can harness that energy if you have another transmembrane protein that capitalizes on the sodium flux into the cell and uses that energy to drive glucose into the cell interior. Okay, so if there are two molecules or a molecule and an ion that are moving in unison, we call it a cotransporter. The jargon doesn't really matter so much. But what you want to really understand here is that there's a source of energy that's coming from sodium potassium ATPase, and then it's used by another protein. And then once the glucose is in the interior of the cell, where the concentration is quite high, then chemical diffusion again can drive the glucose from the interior of the cell now through the basal membrane, where it can join the other molecules in the interstitial fluid. Okay, so let's just summarize what we've established here in terms of energy. We've got sodium potassium ATPase. It's burning ATP to create the gradient in sodium. It also creates a potassium gradient, but that turns out not to matter for this story. We call that primary active transport, okay? And that term refers to the fact that this protein hydrolyzes ATP directly. Our symporter, which is moving both glucose and sodium into the interior of the cell, is taking advantage of that source of energy. It doesn't hydrolyze any ATP itself, but instead it's doing what we call secondary active transport. Okay, it's active because it's going to require energy. It's just that the energy is coming from a different source. It's coming from the concentration gradient from sodium potassium ATPase. finally, we have facilitated diffusion that's just essentially a hole that is specific to glucose, allowing the glucose to move down its concentration gradient. And that's on the basal surface. Okay, so these are all examples of what we call carrier proteins. They all go through conformational changes. You can see here the carrier proteins sometimes can hydrolyze ATP, sometimes they're doing secondary active transport, so using energy from another source, and sometimes they don't require any energy at all. So this provides three distinct ways that carrier proteins help control molecular flux. Okay, so we can talk in more general terms in response to this question which we laid out at the beginning, or at the beginning of this lecture, which we started on in the last lecture, which has to do with control. How do we control and how do we harness these driving forces of molecules? We have carrier proteins providing one class of proteins that helps on this front. They change as conformation change to move molecules, and we can categorize them by how they use energy or don't use energy. I'm less concerned about whether you remember, that it's a glut transporter or that it's a symporter or an antiporter. These are terms that are less important, I think, than how they're using energy. Okay, you really need to understand the difference between primary active transport, secondary active transport, and facilitated diffusion. Okay, so that's carrier proteins. We also have channel proteins. Okay, channel proteins are all driven by facilitated diffusion, and they move molecules through a water-filled opening. So we talked last time about potassium leak channels is just one example, and we're going to see lots of examples of channel proteins providing a really key physiological function. And so there are two classes of these kinds of proteins. One are just always open, like the potassium leak channels. And then the other are known as gated channels. And gated channels are triggered to open. Okay, so in both cases, they affect the permeability of something, like an ion, across the membrane. But they can be controlled. They can be triggered in various ways, which we'll talk about in a little bit. Or they can just be stuck on the membrane, and where they serve as just a porous hole for that particular substance, like for potassium. So that brings us, are there any questions about that? Yeah?
So what do you mean by facilitated diffusion?
Facilitated diffusion is diffusion that is just allowed to occur because the membrane is permeable to it. And the specific way that it's made permeable is by having a channel protein that is specific to that substance. Yeah. Yeah.
Well, is there any, where does the ATP come from first? And is there any other place it comes from other than the glucose symporters?
Okay, so the question is, where does the ATP come from? That's an excellent question that we're going to get to later. But it raises an important point, which is that we're going to see how ATP provides the chemical source of energy in the interior of a cell. It is the currency for anything that requires energy. It is the fuel for intracellular processes. Through your body, we don't shuttle around ATP, like ATP doesn't circulate around through your bloodstream. Instead, we send glucose and fatty acids and other energetic molecules. And then once they're on the inside of the cell, they are converted into ATP. And we'll get into the details of the metabolic processes that allow for that, but it's a good question. Any other questions? Okay. So that brings us to a clicker question, which is, what would happen if sodium-potassium ATPase stopped working in a digestive epithelial cell? Now, this is not an unusual kind. I'll give you the possible answers in a moment. That's not an unusual kind of question that I'll pose on tests, where we might not have talked about a perturbation of a particular system in lecture explicitly, But my thought is that if you really understand how the process works, then if we perturb it, you should be able to predict the outcome. So it's useful when you're studying to, after you sort of run through a physiological system, to pose the question to yourself. What if I were to manipulate this? What would I expect would happen as a result of that perturbation? Okay, so A, the concentration of sodium in the cell would begin to increase. B, the sodium glucose carrier protein would decrease in its activity. C, the concentration of glucose in the cell would begin to decrease. D is all the above, and E is A and B. So take a couple minutes to think about that one. I'll start the software as soon as I can find my cursor. Okay. Okay, give me your final answer on this one. Okay, so, all right, so if we disrupted sodium potassium ATPase, we're no longer pumping sodium out of the cell and into the interstitial fluid or on the apical surface of the cell. And so we would expect an immediate effect on the concentration of sodium. But we're also removing the opportunity for secondary active transport as well, because it's that gradient that is driving the symporter that brings the glucose into the cell. And so B is also a decent answer. And this is a dynamic process. If you stop doing that, then you would expect that then you're going to have less glucose in the interior of the cell, and so its concentration would go down. So half of you got the right answer. There's a question? Mm-hmm.
So that's when we conclude because the symporter and the ATPase are dependent upon each other?
The symporter is dependent on sodium potassium ATPase for sure. Not so much the other way around. Sodium potassium ATPase could care less whether, well, it needs to have ATP, and so yes, that's a good point, that if it doesn't have glucose available, then it's going to have less ATP.
Can you get it from other sources via like metabolism or something?
That's possible, yeah. Okay, so occasionally when I have a gap in my prepared lectures, there's a question over there? Yeah.
Why is the glucose decreasing? Is it moving fat out of the cell, or?
Why is the glucose decreasing? Let's jump back to that. Okay, so In this situation, like in a lot of conditions we'll consider, we sort of draw it statically, but you got to imagine that it's a very dynamic process. And so as soon as you sort of shut off one source for something, then you have the potential for sort of a cascade of responses. So in this case, we're eliminating the opportunity to bring glucose into the cell, and we're leaving open this facilitated diffusion that's happening on the basal surface. Okay, so that alone, if you knew nothing, if there was nothing else going on in the cell, then you would expect that now the glucose in the interior of the cell might reach equilibrium with the interstitial fluid. In reality, cells are more complex, and you can have compensating responses. And then you also have the factor of metabolism and the burning of glucose inside of the cell. So things can get more complicated, but just sort of given our very simple model, then you would expect a drop in glucose. That seems like a fair prediction. Yeah. Okay, good question. Okay, so quite often there'll be a break in between my slides and the presentation due to the timing. And so since on this lecture, so don't go anywhere, we're not going anywhere. On this lecture, we began last time, so now I'm gonna do a break between slides. But what I'm gonna do, the reason why this matters is that when you tune in on the video recording, I try to keep the video recordings changing with the slides rather than being fixed on the date. So it takes me a second to restart the video recording. So just bear with me a moment while I start the new slides and start the new video recording.